Welcome to our second Digestive Lab podcast, and we're going to start with the stomach and go throughout the intestines. So as we look at the stomach, we're going to be looking at different regions of the stomach as we look at the cardiac region, fundic region, the body or the corpora of the stomach, and the pyloric region. And once again, as we look at this low mag shot, we can recognize the mucosa of the stomach with all of its gastric pits and deep gastric glands extending down to the muscularis mucosa. We have the submucosal layer and then our muscularis externa. So again, as we get closer and closer look at the stomach, we can again see our pits that extend from the surface down to approximately this region. And then we can see these long tubular glands that comprise the gastric glands. And this is going to be in the body or the corpus of the stomach. And you can see how they extend all the way down through the lamina propria down to the muscularis mucosa. Now we've turned this on the side, so a little bit different orientation than we're accustomed to. Here we have our surface lining cells, and these are going to be the ones that are going to produce the visible mucus. This is the one that protects our cells from degradation against the hydrochloric acid. These are going to go into the neck cells, which are again mostly our lining cells that are present. And then as we get deeper, we're going to see these larger cells, and these are going to be more of the parietal type cells that are producing our hydrochloric acid. Now, as we get down toward the base, once again, we're coming down in this direction. We can see parietal cells, nice large rounded cells. We have these vesiculated cells. These are going to be our chief cells, our zymogenic cells. Remember, we also call parietals oxyntic cells. And these are going to be the cells that comprise much of what we see in the base of our gastric glands. Now, in this particular illustration, I'm having a difficult time picking out any that I would classify as enteroendocrine, although this cell looks different. It's not nice and round like we typically see with our parietals. It's not vesiculated like we see with our chief cells, so likely that's going to be a enteroendocrine, possibly an enteroendocrine here. Here's another section through a different portion of the stomach. Again, we can recognize the glands. We see the mucosa, submucosa, and a thicker muscularis externa. And this is going to be more down toward the pylorus of the stomach, although we will see some of those differences as we look closer at the mucosal layer itself. Here we see a number of cross sections through our gastric glands down in the lamina propria portion. We can see our pits extending downward. Can't get a good look yet at the cells that comprise this, although we're going to look much more closely. Once again, on the side, we have our surface lining cells, neck cells. Here we're getting into our larger parietal type cells as we're moving down toward the base. This is all lamina propria, remember. Now, once again, down in the base, we have our chief cells. We have our larger round parietal type cells, which I would consider some of these parietal cells, but we're going to have a lot of enteroendocrine. See, this one is smaller. These cells are smaller. So these smaller type cells down in the base, these are going to be our enteroendocrine cells. See, for me, that one's just a little bit big for enteroendocrine. I still would think maybe they mislabeled that one. Still looks more like parietal to me. Right, now we're going to look at a different stain that's really picking up the mucus granules. This is going to be the dark staining material that we're going to be looking at. So here we can see all of the cells that are involved in producing the mucus, whether we're talking about the visible mucus or some of the more goblet cell type lubricating mucus. That's what we're seeing with these red granules in this red stain picking up all of the mucus material. Again, looking more closely there, you can see all of the mucinogen granules that are present in these cells, and you can see the mucin and the mucus that's at the very top, the apical surface of these cells. Just illustrating all of the mucus material that's being produced. And again, remember the visible mucus from the surface lining cells are protecting your stomach and the cells from autodigestion. Now, as we get to the end of the stomach, this is where we're going to transition from our gastric glandular epithelium, which we see on the left. We've got our gastric pits, gastric glands, and we can see the muscularis externa is extremely enlarged in this region, and that is going to extend up and be part of our pyloric sphincter. Now, as we change our mucosa, notice we're changing the mucosa in this region to look like this. This is going to be our intestinal villi. A lot of these white spaces are going to be our central lacteals. But also notice in this region where we have our lamina propria and our submucosa, we're transitioning that. Now we have all these glands. These are Brunner's glands, and so we know we're moving from stomach, pyloric portion of the stomach, into the duodenum. Now, looking a little more closely, again, we've got gastric pits and glands, sort of a weird section through this, but you can see how we're moving from our gastric glands, this is pyloric sphincter, and we can see the beginning of the appearance of these mucus-producing glands down in the submucosa, being Brunner's glands. Here we can see these large central lacteals in the middle of these intestinal villi. There's a good one there. There you can see a good villi with the central lacteal material in the middle. And then these are going to be our crypts of Lieberkühn extending down, discontinuous muscularis mucosa through here, and our Brunner's glands down in the submucosa. Again, another view of duodenum, very clear Brunner's glands, intestinal villi, crypts of Lieberkühn, all the things we are looking for, all the layers we're looking for in our alimentary canal. Just a closer look at the muscle coming up from the muscularis externa, forming the pyloric sphincter with gastric mucosa on the left, intestinal mucosa, and Brunner's glands on the right. Another view of the pyloric sphincter. Here we have a much more extensive run of gastric epithelium, pits and glands. We can see the extensive muscularis externa, the pyloric sphincter, Brunner's glands. This is sort of digitized. I'm not exactly sure what happened here, but here we're getting into our intestinal epithelium. So just a different view of the pyloric sphincter. Here we have another view of that, showing the highlight here being that of Brunner's glands. It's in the inset box. We'll look closer at that. And there we can see those principally mucus cells. And remember, this mucus is also going to be acid neutralizing mucus, emptying into the duodenum here, into the base of the crypts of Lieberkühn to neutralize any excess hydrochloric acid that remains after coming from the stomach. Now, as we look at this cross-sectional view, once again, we can see two very nice examples of our plicae circularis extending upward here. Here's another plicae circularis coming in. We've got our mucosa, submucosa, muscularis externa. Then we have all of this material present in our submucosa. They're not lymphoid nodules, and certainly you're never going to see a muscularis mucosa that expansive. So even at this low magnification, we're a little bit curious that this is likely going to be Brunner's glands because we're in intestine. Plicae circularis tells us that. We're not in colon because we don't see all of the white goblet cells. So that's how we can deductively reason that in this low mag cross section we're looking at duodenum. Now, as we look at the base of the crypts of Lieberkühn, certainly we're going to have those different cell types present. And once again, in the lamina propria, nice examples all the way around of our plasma cells, just everywhere. But as we get down toward the base, here we see those large red granules, and these are going to be those Paneth cells. And the red granules, recall, are producing and storing lysozyme. Now, outside, these are eosinophils. These red cells. Don't confuse these red cells in the lamina propria with these red cells in the base of the crypts of Lieberkühn. Again, just another view of cross sections this time of our crypts. Very clear. We can see those nice large red granular cells that are the Paneth cells in the bottom of our crypts. Just another view as we continue to look at the Paneth cells in several different slides, showing those nice large granules. All you would need to do would be to see this to be able to determine that you're looking at a crypt of Lieberkühn in the small intestine. Now, we don't know which portion of the small intestine just looking at this, but we know we're in a crypt of Lieberkühn in a segment of the small intestine. Now, again, looking at a different area of this same view of the duodenum. This is where we see one of our plicae circularis. We can see our Brunner's glands, so we know we're in duodenum. And then this extension of our submucosa, we really can't see the smooth muscle that extends up here, but you may have some smooth muscle from the muscularis mucosa into the villi. Sometimes you may have this. It's mostly in the sphincters, the pyloric sphincter and the ileocolic valve, where you're going to see that. But this extensive swelling of our submucosa, that's going to push this upwards into the lumen, and that is our plicae circularis, or our valve of Kerckring. Again, another view in this area. This is showing some of our crypts of Lieberkühn in cross section, but it's also showing some of the ducts and acini that are present in our Brunner's glands. So here we have muscularis mucosa. We know that these are cross sections of our crypts of Lieberkühn because of all of these Paneth cells that we see present. And so these are our mucus producing Brunner's glands, and here you can see one of those extending up through the muscularis mucosa, emptying into, we can't see exactly where it empties, but it's going to empty into the bottom of one of our crypts of Lieberkühn. Now we're looking at a different section of intestinal tract. We can clearly see that we have central lacteals present in our villi, these big white spaces. We can see that we've got plicae circularis, the extension of the submucosa up in here. The submucosa is much thinner. And as we look at our villi, they're going to be fewer in number, and they're actually going to be shorter. I know it's difficult to see in this one, but they're coming more slender. These look relatively fat, but as you look overall, these are becoming shorter and more slender. But the biggest thing that tells us we're looking at jejunum is the absence of any glands. So no Brunner's glands. We know we're not in duodenum. And we know we're not in ileum because we don't see any secondary lymphoid nodules, those Peyer's patches. So it's the absence of material here that clues us in that we know we're intestinal tract, but we are in jejunum and not any of the other two because we don't see those distinguishing features of either duodenum or ileum. So here's a closer magnification view, again showing the villi. Here we have a plicae circularis, and all of these big open spaces here, these are going to be our central lacteals in the middle of our intestinal villi. We continue to look at the mucosa that's lining our intestinal epithelium, and what we're seeing here stained in purple, these are going to be our goblet cells. A little bit different stain than we're used to seeing, but we can see our simple columnar epithelium. You can even see the brush border on these, even at this low mag, but the number of goblet cells that we're going to see, and this is going to be ever increasing as we continue to get further and further down through the alimentary canal. Closer magnification, again, we see our striated or brush border across the surface. Here are our goblet cells, and then our surface absorptive cells as we dive down into a villus. This could potentially be a central lacteal. It's difficult to see in this particular plane of section. Now, as we continue to look at alimentary canal, once again, we see the mucosa, submucosa muscularis. We see the villi, so we know we're in small intestine. Some fairly prominent lacteals here, short villi, so we have a feeling we're not in duodenum, but we definitely don't see Brunner's glands. Now, we also don't see Peyer's patches, lymphoid nodules present, so we are still in jejunum. Once again, with this particular stain, they're staining the mucus on the surface. We can see the brush border, we can see goblet cells, and again, surface absorptive cells. Now, the striking feature in this particular field, or our Peyer's patches, we can see our secondary lymphoid nodules aggregated together here in the submucosa, and that's our first indication that we are looking at ileum. Then we look at our thin, slender villi. That's going to be another indication that we're looking in ileum. As we look at these closer, we're going to see an increasing number of goblet cells, and altogether speaks to the fact that we're in the ileum portion of the small intestine. Here at higher magnification of the villi, you can see the number of white spots that we see on the sides of the villi. Those are our goblet cells, and see how slender these are. Much more thin than we saw in either the jejunum or in the ileum. You're still going to have the same components. There will be lacteals in the center of these, not quite as prominent. We'll see those crypts of Lieberkühn with our lamina propria, submucosa, and muscularis externa. Again, a nice view of ileal villi. And again, we can see all those components extending down. We can see a nice view of our crypt of Lieberkühn as it's coming in, even branching out at this point. And then the goblet cells on the surface. And there we have a nice example of a lacteal in the middle of that villus. Now, these secondary lymphoid nodules, we can describe them exactly how we could describe them in a lymph node, for instance. We have the germinal centers, we have the outer coronal material. So these are secondary lymphoid nodules. Again, as we've mentioned before, these are permanent structures that are present in the ileal submucosa and extending up into the lamina propria. So in the villi, in the lamina propria, we're going to see quite a large accumulation of lymphocytes extending up and away from these secondary lymphoid nodules, and many of these dark nuclei that we see staining in the lamina propria part of the villi, even extending up and being in between our columnar epithelial cells, these are going to all be lymphocytes. Here's another view of ileum. We can't quite see the thin, drawn-out nature to these villi, but we definitely see our Peyer's patches. We can see some placae circularis. Again, we know we're in intestine. We know we're in small intestine because of the villi, and because of the secondary lymphoid nodules, we know we're in ileum. Just a higher magnification look, we can see our intestinal villi, crypts of Lieberkühn. Here we can see our muscularis mucosa. We can see our submucosal connective tissue and these secondary lymphoid nodules clustered together in the submucosa. Now we're moving into colon. We basically lose the villi that are present in the intestine, and what we're left with are just the crypts of Lieberkühn that are going to be lined and very highly substituted and filled with goblet cells. So now we're going to examine a little bit of our colon type tissue. So we have the same cell constituents. We have our surface absorptive cells. We can see our goblet cells. We've got our brush border at the surface, our microvilli. That is going to remain unchanged. But the number of our goblet cells are going to be increasing drastically. And you can see in this particular field, tons of goblet cells, goblet cells, goblet cells. In this one, you don't quite see as many. But mostly what we're going to be seeing, rather than our surface absorptive cells or these columnar type cells, are going to be goblet cells. Another characteristic feature of colon are going to be our taenia coli on the outside. That external longitudinal muscle is going to be broken down not into a layer any longer, but into our ribbons of smooth muscle. And there will be 3 of them present. Just a higher magnification look. Here we have our circumferential smooth muscle, and this is our longitudinal bundle or ribbon that is a portion of the taenia coli. Now, here we are at the end of our alimentary canal. Literally, we have our anal canal coming down, and we have the skin on the left-hand side. And so we're going to look at this junction or transition zone between the outside of the body and the anal canal. We have an internal anal sphincter comprised basically of our muscularis mucosa, and our external anal sphincter, that's going to be skeletal muscle. So as we look at that transition, we can see our crypts of Lieberkühn. We do have some lymphoid nodules. These are not going to be indicative of like we see in ileum, not pirate patches. These are not permanent. But we're going to transition from our crypts of Lieberkühn. Now we're getting into our stratified squamous, thin skin type of epithelium. So this marks that transition between the anal epithelium and our surface epithelium. Once again, as we're looking at that transition, this is a little bit on its side. Here we have anal, crypts of Lieberkühn. Here we have our stratified squamous epithelium. This is going to be our internal sphincter of smooth muscle, and then we'll have our skeletal muscle, external sphincter coming in this direction. And there, just another look, seeing characteristic features that we would see of thin skin, much like we saw in our lip before we got to the vermilion zone of that transition. This marks the transition at the opposite end of our digestive tract.